Okay, so in the interest of time, we're going to start. We're very pleased to have Jon Mentxaka coming from Spain to speak with us. He is in the lab of Zorta-Santin, and we are really, really quite interested in his poster and his work that he just presented successfully at the IDS 2024 that was hosted in November in Bruges, Belgium. So a little bit about Jon. He graduated from the University of the Basque Country, UPV-EHU in 2019 with a degree in biotechnology. While in the fourth year of university, he participated in the study abroad program at the University of North Carolina at Chapel Hill. And by the end of his university degree, he was participating in a regional academic excellence program. And he was an intern studying at the Biofisca Institute of UPV-EHU. At the end of his degree project, he concluded some research about DNA repair promoters in A basic sites, and the findings were later published in a scientific paper in DNA Repair Journal. Following an academic focused career, he completed his master's degree on biomedicine and molecular biology at UPV-EHU, and he was awarded a Young Researcher Grant founded by CSIC for another internship. He did research on voltage-dependent potassium channels and epilepsy. Once he entered his doctoral program at UPV under the supervision of Dr. Zorta-Santin in the field of non-coding variants in type 1 diabetes with a special focus on long non-coding RNAs. They actually established a collaboration with Dr. Eddie James' lab at the Benaroya Institute of Seattle, where he actually was in residence for 4 months investigating a new and unexplored question, which is, can long non-coding RNA genes produce peptides which may function as novel autoantigens in the context of stressed pancreatic beta cells? Thank you so much for joining us. This is a very interesting topic, Jon, and we're really excited to hear you walk us and our larger audience through it.
Okay, thank you very much, Monica, for your very kind introduction. And as you said, in the next few slides and minutes, I will try to explain what I have done in collaboration with the Eddie James Lab of the Benaroya Institute in the 4 months that I stayed in Seattle between March 2024 and July. And as you said, we tried to address this question of whether these long non-coding RNAs, which are not supposed to be translated, can actually encode for peptides that can even be contributing to type 1 diabetes pathogenesis. So let me start. And I think it's appropriate to start with a little overview of what long non-coding RNAs are. In brief, long non-coding RNAs are pretty much very similar to regular protein coding genes, with the main difference that they do not finally produce a protein. They are not translated. So for years, they have been considered, or better said, they were considered as part of that junk DNA, junk genome that wouldn't really do anything. But as science has progressed, there's a lot of research now and evidence that show that these long non-coding RNAs, which by arbitrarily are considered to be those genes that give rise to transcripts longer than 200 nucleotides, well, they have been described as actually important because in type 1 diabetes, for example, and in many other diseases, SNPs that are associated to those diseases in GWAS studies, they lie in these long non-coding RNAs. Well, for example, here in this slide, I have shown you some of the few functions that these long non-coding RNAs can serve, which usually are related to gene expression and chromatin remodeling, most of all. They act as a scaffold. Sometimes they can even act as microRNA sponges. They have several functions, and it really depends. The thing about these long non-coding RNAs is that they are not expressed at the very high levels within the cells, but they are very cell type specific usually. So let's keep going.
Jon, can I just ask a quick question?
Yeah, sure.
What levels are they expressed at? And is it temporal in nature? Just to kind of unpack that a little bit.
So it really depends. The ones we have been in our lab, we're interested by long non-coding RNAs, and my close experience, the ones I have been researching, they are expressed at not very many copies per cell, maybe between 1 and 10, but there's some other long non-coding RNAs, let's say she's, which are much more important and are much more studied, and they are expressed much more. She is the responsible for X chromosome inactivation, and it's a long-coding RNA. So it really depends, but the ones that I have studied, they are not very expressed like in very high levels. So we call them long-coding RNAs because, as I said, according to all the predictions that bioinformaticians have done, they don't have the typical characteristics of a protein coding gene. There's some features that basically allows bioinformaticians to predict whether a gene or a feature in the genome will encode a protein or not. And long-coding RNAs usually they don't have them. But anyway, the mRNA that is transcribed from them, it looks pretty much like a regular protein coding mRNA. It has a cap, it has a poly-A tail. Well, it can have it, not always. So that's why no one really looked at them too much. But from 2010 on, when the say revolution or the development of ribosome profiling made a breakthrough, which ribosome profiling allows to sequence those fragments of RNA which are protected by the ribosomes. So they are usually being translated actively. So when these new techniques developed, and people started to see that, hey, there's in this sequencing, RNA sequencing from ribosome profiling, there's actually quite a lot of reads or quite a lot of transcripts that come from non-coding RNAs, not from regular protein coding RNA. So they start to focus more on this and give more attention. Now there's plenty of papers, as I said, from 2010 to currently, that show and have shown that many of these non-coding RNAs actually, maybe not all the time, but at least under some circumstances, can give rise or can encode these peptides. Sometimes I call them micropeptides because the open reading frames found in these non-coding RNAs are usually not very long. As a threshold, we say that under 100 micro amino acids, we call it like a micropeptide. So they're usually fairly short. But yeah, there's plenty of evidence, and even in type 1 diabetes, there's some micropeptides described, at least maybe not directly type 1 diabetes, but in beta cells, some functional micropeptides that come from long non-coding RNAs. So we were really interested by this. Here I show another slide in which I want to illustrate some of the some functions that some already described micropeptides can have, and they can range from JAK signaling to mTOR to even antigen presentation. So here I think that these balls with colors are, I just showed them, these are the micropeptides, but there's a lot of literature now. So going into our hypothesis of these micropeptides and what they might be doing in type 1 diabetes pathogenesis, we were thinking that maybe in the context of the initial stages of type 1 diabetes, when there's no symptoms, but the triggering events occur when the immune tolerance is lost, this leads the beta cell to start a chronic inflammation that it cannot escape. So when the beta cell is even more stressed than it already is in basal state, because it has to produce a lot of insulin, we think that maybe mistakes can happen, and these non-coding RNAs in this beta cell stressed and inflamed state, we think that maybe they come into contact with the ribosome more often than they should, as other ribosome profiling experiments have shown. We can think of it maybe as mistakes that the beta cell makes when it is virally infected and the triggering event has happened and everything starts to go south. So this is why I put here the line that I don't think in my opinion, this long coin, the translation of long coin RNAs would not be a triggering event in type 1 pathogenesis by any means, but maybe these non-canonical or alternative peptides that come from long coin RNAs that shouldn't be there, that we can take as mistakes, maybe they can be contributing to worsening the inflammation and the immune attack to the beta cells.
So maybe not triggering, but contributing.
No, I don't think it's
contributing.
trigger. Yes, exactly. So that's the view we have of it. And well, this can happen. This can keep occurring. I don't know until, well, of course, here the stage 5 will be where the type 1 diabetes is diagnosed. So working with this hypothesis, this is what I was mentioning. Maybe there's a viral infection. This is a very neat image from a review by Decio. And this is why we propose, upon this viral infection, there might be some kind of dysregulation of translation where these long-coin RNAs start to be translated, start to produce these peptides that are unknown to the cell, let's say, and they start attacking it. This can happen in many ways because there's obviously MHC class 1 and 2 presentation. In my collaboration with Eddie, we have focused on MHC class 2 presentation because there's some evidence also, there's some papers out there that suggest that beta cells upon interferon gamma induction, they can even start expressing HLA type 2 molecules. So this is what we want for. This is our diagram, working diagram. I hope it's clear. So what have we done? First of all, when I was still in Spain, what we did was take endo-C beta H1 cells that we work with, treat them with poly-IC, which is this molecule. Oh, can you see my pointer? I guess not. Can you see it now?
Yes,
Yeah,
I
right?
do see it. Yes, fine.
Okay, so this is what would be a viral mimic, what we use to cause the inflammatory response in the beta cells. And what we did pretty much was pull down by an affinity method the ribosomal complexes and extract all the RNA associated with them. The drawback of this is that when you pull down everything within and around the ribosome, yes, you do get what is being translated, but you can also get some transient RNA molecules that are by chance next to it, or even regulated translation. So you get a bit of a mixture. But this was our working model. So once we purified that ribosome-associated RNA, we performed regular RNA-seq. So we had, let's say, just a typical volcano plot in which we had upon the inflammatory stimulus, we had just a series or a list of genes, long-coin RNAs, which were upregulated. In this case, upregulated means that they came into contact with the ribosome more often in the stimulated condition with respect to the baseline one. So this was all these genes are a good starting list to try to now search for those that have actually good features to be translated, but there were a lot. Between them, I want to mention this one that had already been published not long ago as protein translating, even if initially it wasn't. So we were on the right track with it. So since this list was quite big to work with, we decided to go for a multi-omic approach, let's say. We did perform the same experiment, but this time, instead of purifying the RNA, we purified the nascent peptides with another protocol. And then we performed mass spectrometry. The goal of all this is to converge, and let me walk you through this. With the RNA-seq that I had done, I had a list of many long-coin RNAs that were associated with the ribosomes. What I did was take all the open reading frames of those sequences, translate them, and obtain a list of, let's call it a list, a dataset of potential translated products of long-coin RNAs. With this, well, we then perform the mass spec of the nascent peptides of the ribosome. And what we did is use this database that I built custom to match with this spectra from the mass spec. This way, we obtained a list of candidates, some matches, not many as one would expect. At the end of the day, they're long-coin RNAs. Even if some do get translated, it shouldn't be the majority. And with this list that I obtained, this is where I started my collaboration with Eddie, and this is when I went to Seattle. So, wait, why can't I? Oh, there we go. So this is the overview of what I did in collaboration with Eddie. Eddie James, sorry. Those, first of all, as we had a few candidates to explore, we ran a prediction of, in this case, HLA, the type 1 diabetes HLA risk allele. In this case, HLA DRB1 0301, because it's quite a prevalent allele here in this population and associated to type 1 diabetes risk. We ran this prediction to find the best within those peptides, to find the best epitopes that could potentially bind to this molecule. And this was done in the computer by Eddie. And the top 40 candidates, we synthesized them. And the next thing I did over there was to perform an actual binding assay, a competitive binding assay. The binding assay allowed me to select, again, the best candidates to keep narrowing down our list. And those best, I think I took more or less the top 20 candidates, which showed some good, or at least some binding to some extent. Those are the ones that we assayed or we incubated with the type 1 diabetes patient PBMCs to then perform CD4 lymphocyte activation assays. So let's go with the competitive binding assay. The way this works, the results, the way this works is you have the monomer of the HLA of interest you are going to interrogate, and you have a reference peptide, which we know for sure it binds to the monomer. And what we want to see is a displacement of this reference peptide that already binds and gives a signal at the end of the assay by the peptides we are interrogating. In this case, I named them just from P1 to P40. So as the graph shows, the less the signal intensity, the higher the binding it is. In this box, I just have, well, I can't even see the box. Okay, in this box, there's just a summary and the pooling I did of all the peptides I selected. So I pulled together the strongest ones, and then the next five. So because it's too many to try, I don't have as many PBMCs to try with individual ones. So you have to pull them at the beginning and then search for individual responses. I pull them together according to their binding strengths. So one, maybe a moderate binder, would not be masked by the effect of stronger binders. So this way in bold, you have all the peptides that we selected to carry on with in vitro assays, which were around 20, pulled in four pulls. Next, after this, the in vitro experiment started. You have the timeline, top left. We start with the type 1 diabetes donor PBMCs that very kindly Eddie James Lab provided me. And we treat them with the pool of peptides, either pool 1, 2, or all of them. You leave them be for 7 days a week. And after that, we added IL-2 every other day, to drive CD4 T cell proliferation. At the end of this process, around 2 weeks to 20 days, we perform what is called a CD154 AIM assay, which pretty much is an assay to check for activation of populations of CD4 positive cells. And in the lab, in these panels, this is just the strategy for the gating. First, we select, in these 3 first panels, we select the live cells and single cells. And in the last panel, we select for the CD3, CD4 positive cells. And on the right, there's the examples after this gating of what the graphs look like. So as a positive control, because we didn't really know if we were going to find any positive results for our peptides, we have a tetanus peptides, very well known. It's well known to drive activation. And okay, let me move this. And this is how an activation positive response look like. There's a population very clear in the quadrant 2, which shows it in the red box. And a negative response would be on the bottom, just no difference when we treat it with DMSO with respect with that when we induce this AIM assay with the peptide. So surprisingly, or maybe not as surprisingly, these were our results for the assay. I performed in my stay there more or less 12 experiments with different type 1 diabetes donors. And these peptides, P10, 37, 17, 21, and 39, consistently showed positive responses. What is consistently? For instance, for peptide number 10, I would say that out of the 12 or 13 trials that I did, in 6 of them, remember that each trial is a different type 1 diabetes PBMC donor. So it's a different individual. So out of the 12, at least 6 of them showed positive results. This is just an example of one of the trials, but we could see these positive populations quite often. So this was exciting. The next steps, according to, in the methods that Eddie James Labs developed, is to try to generate lines from these assays. So this is just the initial screening after the in vitro expansion of the CD4 cells. What you do afterwards is try to generate lines. So you sort those cells. So you put on each well just 4 or very few cells to try to generate a line which is almost exclusively activated when you treat it, when we spike the peptide in. So in the top right, you have the process of the we have the process of the line generation, and at the end of the process, we made sure we checked again with the same assay, the AIM assay, that these cells were still positive, as you can see, compared to before, which most of the cells were still not activated. What you get after the generation of the lines is very beautiful, very beautifully activated lines. These are examples for P21, 37, and 39. So the next question we tried to answer in my 4 months there was, okay, these lines that I have generated, we have started all this by using the HLA DRB1 or 301. Are they really, is this activation really driven by presentation of that HLA molecule? So what we do is what we call a proliferation assay, in which pretty much, sorry for the overlap there, my bad. What we do is we incubate irradiated APCs, which are restricted to, which only express this allele of interest, DR0301. We incubate them with our positive CD4 lines that we just generated. We incubate with a peptide, we spike it in, and after waiting 3 days, we add thymidine, the radioactive thymidine. And then at the end, some 18 hours later, we count the radioactive counts with the scintillation counter. Then what we do is the division or the quotient between the peptide stimulated mixture with that had DMSO, the control. If that ratio is higher than 5, we can say that that line that we generated is in fact restricted to that HLA molecule of interest. And here I want to show you that all the lines that I had generated, for P16, which is this group here, and 17, we didn't have any lines which were restricted to that, unluckily. But for the other, we did find some. Well, for P39 also, we didn't find any. To reinforce these results, we took P21 and P37, and we tried to do what is called a tetramer staining assay, which also reinforces the HLA restriction that we have observed. The way this works is that we produce a tetramer, which we load, well, a tetramer of interest, which we load with our peptide. This is done in vitro. And then instead of staining for CD154, let's say, we stain with this tetramer, the cells or the lines that we have generated. In the left, I put an example of how a positive tetramer staining would look like, very clear staining here in the quadrant 2. For P21, we did manage to find or to observe some staining to some degree, not the strongest one, but this, together with the results from the previous proliferation assay for P21, show that this line, in fact, we can say firmly that it is driven by HLA-DRB0301 restriction, T1D risk allele. For P37, the results were not very clear for the tetramer assay, but we will keep working on this. So as a summary, here I have a table, very simple table, with all the peptides that showed consistently positive results in different type 1 diabetes patients, the lines that I generated from each of them, and how many of those were actually restricted. And the interesting thing in all this is, well, this is fine, but I have only talked about peptides, but I started my talk talking about non-coding RNAs, right? So where do these peptides come from? And very, well, I don't know, yeah, very surprisingly, at least, well, I don't know if surprisingly, but interestingly, these peptides, 3 of them converge into a single non-coding RNA gene of origin, a single transcript. So in a way, it does reassure, or it does show us that maybe, in fact, this non-coding RNA3, it is being translated, because if from just peptides we have 3 pointing towards it, that this could be a thing. Well, this is definitely a thing we will try to keep working on, but this is exciting stuff. Same thing with non-coding. Okay, I messed up the colors here, but non-coding RNA1 had also two of the positive peptides, 21, and the 13 came from non-coding RNA1. And finally, another one from the other. Sorry for the mistakes with the colors. So this is more or less, well, this is pretty much what I managed to do in Seattle and what I explained in my poster, and Monica very kindly invited me to explain today. I hope it was clear, but yeah, I hope you enjoyed it.
Thank you, Jon.
Oh, sorry, my conclusions, I almost forgot.
Yeah, I was going to say, do you have a conclusion?
Yes, sorry.
No worries.
So yeah, upon peak exposure, there's an increase in non-coding transcripts, as we saw in the volcano plot. But again, that has to be finely tuned, because in the protocol that we did, we not only get the ribosome protected fragments, also some other stuff. So, but this is a conclusion. Then, for sure, the combination of the RNA-seq together with the mass spec, together with prediction potential, coding potential predictors that are available online and described, that they for sure increase the chances of having very good data and a very good list of candidates to start working with. Not only because if we only do maybe RNA-seq or ribosome profiling, you might be missing what is really happening with the peptides, So the combination is obviously the best. And what I learned from Seattle is that based on these T-cell activation assays that I showed, there are, in fact, in type 1 diabetes patients, CD4 T-cells that recognize these peptides, these long-coding RNA-derived peptide epitopes. And not only that, but these peptides, at least in some cases, are indeed being presented by the type 1 diabetes risk molecule, in this case, HLA-DRB0301. What we're currently working is in the confirmation of those non-coding RNA 1, 2, and 3 that I mentioned in the last table. We're trying to actually show proof of direct translation in beta cells now. And talking to Eddie James, it is sensible to think of similar projects with different type 1 diabetes risk HLA alleles, like PQ, even 0401. So this is work in progress, and we will be working on answering these questions. Thank you very much. Yeah, now I'm done.
Thank you, John. I'm gonna open it up for questions. So yeah, this is really an exciting collaboration between, Isorta Santon-Gomez and in the lab here, and between your lab in Spain and at the Benaroya Research Institute in the lab of Eddie James. It's really a unique opportunity and kind of a very interesting hypothesis. Oh, here's a question. Did you determine whether stress induces any expression of LncRNA expression?
Yes. Upon poly-IC infection, our viral mimic, there is plenty of long non-coding RNAs that are induced and upregulated. The thing in what I showed is what I did was at the RNA-seq of what is associated to the ribosome. So we are kind of in that volcano plot, we're only looking at that fraction, which is not what happens in the whole cell. But for instance, I haven't shown data of this because this is what I have done in Spain. But some of those long non-coding RNAs that appear in my volcano plot of ribosome associated long non-coding RNAs, I have been working with one of those, and upon stress induction, it is upregulated, not around 1.5%. And actually, I have a strong evidence that that one encodes a real micropeptide, and I'm working with that.
Another question, do LncRNA's 3-to-1 peptides are LncRNA 3-to-1 peptides specific to beta cells, or could they be expressed on other cells also?
Since this is like out of the oven kind of, I have not checked. I do know which the LncRNA 1 and 3 are. I know their sequences and names, but I have not checked in other lines or cells for their expression. I believe that I don't see why they could not, but it will really depend, because as I said, long-coding RNAs are usually quite tissue dependent. So in this case, I'm working with beta cells, and also not any beta cell. It's a endo-C beta H1 cell line, which is, well, it has its own characteristics, but I don't see why it couldn't be expressed in other cell types. I cannot answer that for sure.
Yeah, no, that's fine. It's just a nice thought experiment to kind of work through. There's a long history of sort of this long non-coding RNAs, the involvement of long non-coding RNAs in the pathogenesis of autoimmune diseases. I mean, there's a series of papers out of China, one as recent as 2020. And so I think it's just a really a sort of a darkened corner in type 1 diabetes that you really have had the opportunity and excellent training to shine a light on here. And it's really exciting times. It will be really interesting to see where this work goes, John, and we'll be following closely to see what's next for you. Jemming Liu, do you want to unmute and ask a question?
All right. I want to open the two channels to listen out loud. So can you hear me, right? Can you hear me?
Yeah,
Yeah,
Brian, I can hear you.
you.
It's a little bit garbled,
Okay.
but yeah, go ahead.
Sorry. So
There,
I have
that's
to
better.
use my cell phone. Sorry. So
Okay,
I
much
have
better.
some questions because recently our lab also worked on the LncRNA. So first question about when you treat the endo-C BH1 cells with the poly-IC, what's the mechanism regulating non-coding RNA expression? That's first question. The second one,
So
what's the
Let him take that one first. What's
yeah,
the mechanism?
maybe I didn't understand very well, but what's the mechanism behind what exactly? I couldn't get that very well.
what's the mechanism regulating non-coding RNA expression? Through what kind of transcript factor and also the
Yes,
coming
I'm not
from like okay.
I'm not an expert on that, but pretty much long-coding RNAs act in almost every way as normal RNA genes. So they do have they are under the influence of transcription factors, enhancers, and that kind of regulatory elements. Yes.
Okay, thank you. And the second one, about the long-nuclear RNA become translated into the peptide. What's the mechanism regulating those long non-coding RNA become the coded peptide?
Mm,
Yeah,
that's
how does
very,
it
very.
become the code? How does it become a peptide? Is that the normal route, or is it just under duress and pressure?
That's a very interesting question. How does this initiation of translation even start for the case of long-encoding RNAs, which don't have these typical features of normal messenger RNAs? And I'm not sure. We have not explored that for the moment, so I don't think I can answer that. I don't know if it is an
No worry. I still
dependent mechanism, or I don't know if there's some other ribosomal entry sites. I'm not sure.
Okay. So and also the third question about I will cross compare the peptide coming from the long non-coding RNA with well-known autoantigen. For example, the insulin or the GAP65. I mean, so have you cross compare your peptide with the autoantigen in the beta cells?
Nice.
So if maybe Monica, you can help me if I understood correctly, if I can cross compare them, or
Did you cross compare the sequence of your epitopes with any of the known autoantigens, autoantibodies,
no,
insulin 9 to
with
23,
GAP6,
or
we have not done that. We have not cross compare to check for similarities, I assume, or in sequence.
So
And
yeah, the reason I asked those three questions because they're all coming from the last one. So you only list the LncRNA 1, 2, 3, 4. What's the real name of the LncRNA? So the reason, for example, in our labs, we do have several LncRNA, the NEAT one, and also the one I'm working on. I just really want to know what's the LncRNA you are doing right now. So that's really the fundamental question. So the user will give us the real name.
The reason why I have not put the real name, well, first of all, they don't have a name, these LncRNAs, because they are not. They do have how do you say it? They do have an ID or ENSG identifier, but they do not. Most of them don't have a name, and even if they did, I have been suggested by my supervisors not to unveil their names yet, in case case we do some further research.
Well, it sounds like maybe an offline conversation for a potential collaboration with the Santa lab and the Lou lab in California.
Thank you.
Yeah, that sounds like a great conversation.
Thank you for your questions.
No worries. Thank you. It's a great talk. I love your talk. Thank you.
Thank you. Well, that's really, we're at 12:39, John, and we've had some good questions. Great presentation, really interesting questions that you're asking. And, hopefully you and the City of Hope scientist, Jemming Brian Liu, can speak offline and further that conversation. But congratulations, and it was a great poster you presented at IDS, and we look forward to seeing the next publication that comes from your group.
Thank you very much.
Thanks.
Have a great rest of the day.
Have a great rest of the day. Bye-bye now.
Bye.